Hi, I'm Stephen Becker, Associate Director of the Office of Regenerative Medicine at the National Eye Institute. I have no financial conflicts to disclose. It's my pleasure to introduce today's ARVO special session featuring the NEI Audacious Goals Initiative, New Factors in Regenerative Medicine for Vision Research. The NEI AGI started a 15-year effort to catalyze innovation in vision research. At the start of the initiative in 2013, there were indications that it was possible to restore vision using regenerative medicine approaches. Transplantation of stem cell-derived precursors had restored vision in rodents. Also, the ability of some fish and amphibians to regenerate their eyes offered the possibility to activate similar repair mechanisms in the mammalian eye after injury or disease. Also, methods to reprogram and differentiate cells from adult cells using induced pluripotent stem cell technology had been established. Our audacious goal is to regenerate neurons and their neural connections in the eye and visual system. Our targets for replacement are photoreceptors and retinal ganglion cells. Over the last few years, the NEI has established 3 consortia. The first focused on developing new imaging technologies to enable the in vivo monitoring of activity of individual retinal neurons. These projects have been featured at previous ARVO annual meetings. Our latest consortium had been developing new animal models that are more accurate in reflecting the human visual system and will be used to test cell replacement strategies. Today, we'll highlight projects from our second consortium, which aim to identify unknown factors critical to regeneration of neurons, their axon guidance, and making new functional connections. 6 funded teams employed a variety of approaches to screen, identify, and validate targets that may prove useful in efforts to restore visual function to the visual system. These projects will contribute to a publicly available data repository and will enable additional research to explore related factors and pathways in other animal and disease models. 3 groups are identifying molecules associated with optic nerve regeneration. These collective efforts will provide a better understanding of how proteins, genes, and lipids respond after optic nerve damage. 2 other teams are researching how Mueller glia regenerate retinal neurons in zebrafish and other species, with the hopes of unlocking their regenerative capacity in humans. The 6th team is exploring biological mechanisms that may help to integrate transplanted photoreceptors. Here's a listing of today's speakers and their talk titles. Afterwards, we will have an opportunity for questions and discussion, and I hope you enjoy the following presentations. Thank you for your attention.
Hello, my name is Mike Dyer, and I had the pleasure of serving as the chair of the Executive Oversight Committee for the AGI initiative focused on neuronal regeneration. And it became clear early on that the research teams were going to generate a large amount of next-gen sequencing data, and they needed a mechanism to share that data with each other during the project, and also to share that with the broader community once a project was complete and the studies were published. And so we thought the St. Jude Cloud would be an ideal platform for those sharing mechanisms. And the St. Jude Cloud is a partnership between Microsoft, DNA Nexus, and St. Jude Children's Research Hospital. And its goal is to really address many of the issues with data sharing and credentialing that we face in the cancer field as well as the vision community. And if you're interested in some of the details of how the St. Jude Cloud addresses those issues, I encourage you to read this paper in Cancer Discovery by Clay McLeod. So we developed a landing page for the Audacious Goals Initiative on the St. Jude Cloud, and this is integrated with an existing page focused on epigenetics and retinal development and retinal disease. So it's called the retinal nucleome page. And if you want to find this landing page, you can use this web address here, or simply Google St. Jude Cloud and retinal nucleome. And within this are some previous publications showing changes in the epigenetic landscape during human and mouse retinal development. There's aged mouse retina, as well as disease models like retinoblastoma. We've also included epigenetic data for other regions of the central nervous system, such as the cerebellum, because often it's useful to compare across different regions and see what is similar and what is different. So I'm going to show you a few of the resources that are already available that were previously existing, focused on epigenetics, and then we'll dig into a bit of the Audacious Goals Initiative data sets. So here's one of the epigenetic browsers, and this is looking at mouse retinal development across several stages, including an aged mouse retinal profile. And on the right here, you can see gene expression. This is RNA-seq data. So you can see how the individual genes, in this case CRX, is increased with retinal development. And these colored bars are the integration of ChIP-seq using a mechanism called hidden Markov modeling. And the hotter colors, the reds and oranges, are more active chromatin states, and the cooler colors like green and blue are more silent chromatin states. But you can also interact with the data and pull down individual ChIP-seq tracks or whole genome bisulfite sequencing and zoom in and out at any level of resolution using this browser. And so it's highly interactive. I use it to visualize a variety of genes on nearly a daily basis, and you can use this even to make figures for publications. One of the other challenges we face in the cancer field, as well as the Audacious Goals Initiative, is how do you share and visualize drug sensitivity data? And so we took advantage of a visualization portal developed by Anang Shalat, and this is also part of the AGI. I'm just showing you a cancer sensitivity portal, but it's very similar for the AGI initiative, where you can get a high-level view of sensitivity, maybe across different drugs or across different samples, as shown here. But you can also dig into the actual dose response curves and some of the key quantitative metrics like EC50, area under the curve, and how well the curve fits. And this can be filtered, in this case, by the class of drugs, the particular drug, or the molecular target. And so again, just to quickly look through large amounts of data, this is a very useful visualization tool. But the raw data will also be available for the cloud-based portal for the AGI. So here's what we've put together for the AGI. As you can see, we have the logo. And if you click on this studies link, it'll take you to a page that lists each of the teams that was supported by the AGI. And under each of those teams, you'll see buttons for a data summary that just provides a high-level view of what type of data was generated. A link to the raw data, so this is for computational biologists or for those who want to reanalyze the data, to any visualization tools, and then to relevant publications for each one of these teams. So it's still in beta testing, and we're eager to hear your feedback if you find links or components that are not working, but we think this is going to be a really important tool going forward. And so I just wanted to show you one example. I'm going to go live to the web and show you how we use this. This is a single-cell RNA-seq dataset generated by the Blackshaw group. This was published in Neuron. It's hosted on the AGI portal, and it's profiling mouse retinal development. So let me leave this. Go to the web. So this is going to be a live demonstration. And so here are the data, just as I showed you before, but now we're interacting on the web. And this is a massive data set, but you can visualize this from any computer. And again, you can see the different colors represent different stages of development and different classes. You can zoom in and out and change the size of the dots as needed. But typically what you want to do is look up the expression of your favorite gene. So you just use this drop-down menu and click on gene expression. I'm going to type in CRX. And what you will now see is that very quickly it will highlight the expression of CRX in those subsets of cells. I'm going to make the size of the dots just a little bit bigger so we can see it. And I think that you can see that there are some red dots here. Right here in this area, these are the CRX expressing cells. Now, that's just to quickly visualize, but often what you want to do is see more quantitative measures. And so you can use this drop-down menu to visualize this in a violin or box plot. In this case, I'm going to show it by cell type. And so here's a violin plot, or if you want to switch that to a box plot, you can now see that CRX is expressed. I'm going to exclude the non-expressing cells. CRX is expressed in photoreceptor precursors, cones, and rods, just as you would expect. And so you can do this and even do side-by-side comparisons of different genes using this visualization portal. So I hope you appreciate this. It's very user-friendly, very easy to use, and much quicker than downloading the data and processing it and visualizing it yourself. So to conclude, in support of the AGI, both for researchers during the project and to share data after the fact, we have developed a landing page on the St. Jude Cloud. Again, here's the web address. You can Google search St. Jude Cloud and retinal nucleome to find it. And we think that this is going to be a really valuable legacy going forward to accelerate discovery in retinal research and retinal disease research and retinal regeneration for years to come. And so please feel free to reach out to me or others on the AGI team if you have questions about the St. Jude Cloud. And I'm really excited to hand this over to the individual teams to share some of the exciting results from the NEI AGI initiative.
Hello, I am Sahil Shah, and I'll be presenting today some of the latest work resulting from the Audacious Goals Initiative from the NEI. Our collaboration includes Jeffrey Goldberg and Andy Huberman from Stanford, Larry Benowitz at Harvard, and Holly Klein at the Scripps Research Institute, and we're aiming to enhance optic nerve regeneration using multiple omics approaches. First, I have no financial disclosures. The central broad question we started with is how do CNS neurons respond to injury? Understanding the cellular responses to injury and disease can lead to insights into mechanisms of neurodegeneration, as well as therapeutic targets to improve neuronal survival after insult. Mammalian CNS neurons, unlike their peripheral counterparts, fail to regenerate their axons after injury. This is due to a multitude of factors. CNS neurons intrinsically have low expression of pro-growth factors and high levels of anti-growth factors. Extrinsic cell communications and glia cells in the CNS make more inhibitory and less pro-growth factors compared to the PNS counterparts. And CNS axons tend to form a retraction bulb as opposed to a nascent growth cone. In retinal ganglion cell axons in the optic nerve, part of the CNS, the number one cause of optic nerve degeneration is glaucoma. So why do retinal ganglion cells die in disease and after injury? Understanding the reasons for RGC loss is essential to preventing it. Glaucoma is a neurodegenerative disease associated with increased intraocular pressure and is one of the leading causes of irreversible vision loss. While increased IOP is distributed throughout the eye, the optic nerve head, where RGC axons exit the globe, is particularly susceptible to damage. This loss of vision comes as a result of degeneration of retinal ganglion cells and their axons. And while age is the leading risk factor, increased IOP is the only modifiable risk factor. The molecular changes in retinal ganglion cells occur at several levels. First, at the level of the transcriptome, second, at the level of the proteome, and finally, the transportome, which is the subset of proteins synthesized in the cell body that subsequently travel down the RGC axons to their synaptic targets in the brain. And while our group has developed techniques and explored fundamental biology relating to all three of these levels of cellular dynamics, I will primarily focus today on the work in our changes in the RGC transportome in neurodegeneration. First, however, I want to talk about some highlights from our transcriptomic approaches. In collaboration with the Geschwin lab, we first compared the transcriptomic changes underlying two orthogonal regenerative states. First, in early development, when immature RGCs can extend their axons, and then after optic nerve injury in adult mice that were treated with a regenerative cocktail. After isolating the RGC RNA and analyzing the sequence data with gene set enrichment analysis, we were able to rank the transcriptome factors that were most positively and negatively associated with regenerative states. This ostensibly would highlight the broad regulators of pro-growth states. With further in vivo analysis of our top candidates, we indeed found that inhibiting REST activity or promoting ELK1 activity both significantly increased optic nerve regeneration in adult mice. This has two effects. It identifies putative therapeutic targets as well as validating our transcriptomic approach. Moving now toward the transportome, how are we able to label these proteins that are transporting throughout the visual system? Our strategy involves injecting a biotin compound, NHS biotin, directly into the vitreous space. This tag binds all available lysine side groups and N-terminal amines and has a known molecular weight. Over the course of just a few hours, most proteins in the retina will be tagged, and the subpopulation that leaves through the optic nerve head through the RGC axons can be tracked. These labeled proteins can then be isolated. Using these NHS biotin injections, we can then section and stain these tissues for histology or digest the proteins into peptides for mass spectrometry using a specialized protocol developed by Lucio Schiaparelli, one of my collaborators on these projects. In short, we measure the actual biotin mass shift in each peptide directly. This decreases the identified peptides and proteins considerably, as each peptide must have a biotin-related lysine residue, but greatly increases our confidence in the identified peptide. In these sample slides from mouse retinas labeled with either NHS biotin or saline, we can see highly specific biotin incorporation, which is strongest in the proteins in the ganglion cell layer. As we look further down the visual system, such as in the optic tract and the lateral geniculate nucleus seen on top, and in further detail in the next few rows, through electron microscopy, we can see biotin signal in the RGC presynaptic terminals in the left two black and white pictures, and even sitting on microtubules on the right. And here's the labeled sections. You can see the LGN, which is bright green, and the optic tract, as well as the presynaptic regions, which have more biotin-related proteins compared to the background. So when we identify these labeled axonal proteins, what do we find? We find them to be part of the neuronal portion of the optic nerve, the RGC axons. By using canonical markers of different cell types, we can see undetectable contamination from other cell types, such as astrocytes or immune cells in the optic nerve, meaning that our technique specifically identifies the biotin-related proteins that were tagged in the cell body of RGCs in the retina and then transported to the optic nerve. Taking this next step, we want to be able to model RGC-specific cell death. To do this, we use optic nerve crush, which is a well-established technique. Importantly, many advances in regeneration therapy have used this model, setting us up to directly compare our results with previous studies. In this model, the optic nerve is physically crushed, resulting in degeneration of RGC axons and eventual cell death. With our labeling method, proteins will transport throughout the control eye and optic nerve after intravitreal injection, but should accumulate at the crush site in the injured condition, which we do see on the bottom left. Quantification of these groups is also reproducible, as seen by the Pearson's R coefficient in these replicate groups in the bottom right. We next want to know which specific proteins were changed relative to the total transport. In this volcano plot seen on the left, we have several proteins that were increased in transport and some that were decreased in transport relative to control. One that immediately stands out is KIF5A, which is labeled in red. It's a kinesin motor protein responsible for the transport of other proteins, mRNA, and mitochondria anterogradely towards the axon. We validated this transport reduction through immunoprecipitation, seen here in the middle. Interestingly, the expression of KIF5A was not changed at this time point after injury, just the transport was decreased. We then reanalyzed our original volcano plot, now on the right, for other motor proteins, and did not see the same relative transport decrease, even in a related kinesin isoform KIF5B seen in the direct center. We next wanted to see what happens if you lose KIF5A in an adult mouse. We gave RGC-specific Cre recombinase to adult flox KIF5A mice to stop synthesis of new KIF5A, and we can see the results in this Western blot. In the top row, we see no more KIF5A detectable in the retina or the optic nerve. Interestingly, KIF5C, a sister isoform, does not increase in expression, which was a hypothesis previously put forth in the literature. Immunostaining of retinal flat mounts confirms the loss of KIF5A in RGC axons. We now wanted to go back to our original technique and see what happens with the loss of KIF5A on the RGC transportome in the absence of any other injury. We found deficits in several specific proteins, which actually happen to form a few clusters of related proteins. While more work will be needed to dissect this further, we hypothesize that these proteins may represent larger complexes that are transported down the axon by transport machinery that require KIF5A to function. When we compare the proteins that were decreased in transport in both optic nerve crush and KIF5A knockout conditions, we found an overrepresentation of mitochondrial-associated proteins. With further in vitro testing, we see that the knockdown of KIF5A in RGCs decreases the average velocity of mitochondria and shifts the balance of transport to more retrograde than anterograde, that is, back towards the cell body. Conversely, overexpression of KIF5A increases the average velocity and induces a larger percent of mitochondria to be moving anterogradely towards the axon terminal compared to control. Seeing that we have an effect on mitochondrial function, we then asked to see how the loss of KIF5A affects the visual function of RGCs. We used pattern electroretinograms to isolate the visual response in mice. We found that KIF5A knockout had drastically reduced functionality by 4 months, seen here in these pattern ERG plots. Now, we next wanted to know what the effect of KIF5A knockout was on RGC survival at different time points. We found about a 40% decrease in RGCs at 1 month and almost a complete loss of RGCs by 4 months in homozygous knockout animals. We tested a dose effect by using a heterozygous knockout and found almost exactly half, that is a 20% RGC loss at 1 month. These data demonstrate that the loss of KIF5A results in progressive dose-dependent degeneration. We then confirmed the specificity of all these effects to KIF5A. As you can recall from our original volcano plot, KIF5B was not affected in transport from optic nerve crush. And indeed, when we knocked out KIF5A, we found no significant cell death at 4 months. So we have found a specific transport deficit of KIF5A that results in RGC axonal degeneration. As the expression of KIF5A was unchanged, simple overexpression of KIF5A would not restore transport, as seen on the right. As you can see on the graph, that actually the RGC survival was decreased after KIF5A overexpression with optic nerve crush. We must then develop a method to increase transport of KIF5A and other effective molecules in the face of optic nerve injury. Together, we now have techniques and data on multiple levels of cellular regulation during neurodegeneration. We have the transcriptome, we have the newly synthesized proteome, and we have the transportome. We are now working to integrate these data to form a more complete understanding of the most relevant factors to target for regeneration. For instance, what changes in the transcriptome are reflected in the newly synthesized proteome? And what proteomic changes are missing in the transportome? In addition to KIF5A, ELK1, and REST, leveraging these multiple omics approaches will help us identify the most relevant targets for regenerative therapeutics. All the proteomic data presented in this project, all the newly synthesized proteomes, as well as the transcriptomes, will be available in the central data repository at St. Jude's upon publication. Thank you, and I want to acknowledge Takanoro, Lucio Schiaparelli, Yuquin Yin, and Yuanhui Ma, who worked on these projects presented. On behalf of the Goldberg, Huberman, Klein, and Benowitz labs, we would like to thank ARVO and the NEI for the opportunity to present this work today. Thanks.
Hi, everyone. My name is Jane Lindborg, and much of the data that you'll be seeing today was done in the lab of Dr. Steve Strittmatter at Yale University. And the focus of this project was to identify novel regeneration genes whose loss of function in the central nervous system increases regeneration following a nerve injury. Much of the data that you will see is, it was recently published in Cell Reports, so please feel free to check out that publication if you would like to see the data in more detail. But prior to getting into anything, I and the authors involved in this study would like to say that we have no competing financial or commercial interests. As many of you know, in the central nervous system after injury, expression of certain genes actively impedes regeneration. Now, one such example is P10, and quite a few studies have shown that if you inhibit or knock down P10, you can significantly enhance CNS neurons' intrinsic capacity to regenerate, and regeneration is much better following that inhibition. Now, we aimed to identify a slew of new regeneration genes using an unbiased genome-wide loss of function axonal regeneration screen in vitro. Now, the screen was published several years ago by Steve's lab and evaluated over 16,000 genes for their potential role in regeneration using mouse cortical neuron cultures. So following lentiviral shRNA delivery to each of these 16,000 genes, an injury was induced using a 96-pin scrape tool, and then the neurons were allowed to regenerate their axons, after which regeneration was measured. Now, out of those 16,000 genes, we identified over 400 new regeneration genes, many of which were involved in cytokine signaling, receptor binding, and transport. Now, following this in vitro study, we wanted to determine whether we could recreate the effects that we saw using an in vivo model of neurodegeneration. Here, a mouse optic nerve crush injury model. So we developed shRNA AAVs against each of those individual 400 genes and injected them intravitreally, performed optic nerve crush, and then injected with CTB to label those regenerating axons. And positive axons were those that extended 500 microns distal from the crush site, where the crush site is that accumulation of CTB. A summary of that regeneration is shown in this top graph here. And the red line indicates the regeneration of a non-targeting scramble control. Now, out of those 400 genes, we identified 40 top positive regeneration hits with regeneration phenotypes that were significantly increased over non-targeting scramble control. And over on the left is a summary or a list of those 40 top positive gene hits. And once you have a chance to look at that list more thoroughly, you will find that it is quite diverse. Now, we wanted to validate those in vivo findings using a different system, here a CRISPR-Cas9 gene editing system. And so the experimental paradigm was the same as that with the shRNA AAV, except we injected single guide RNA AAVs into Cas9 transgenic mice, where the Cas9 endonuclease is driven by the CAG promoter. And we were able to validate most of those findings that were determined using shRNA AAV, and indeed found that there were several gene hits with comparable regeneration phenotypes to that of P10. And we also wanted to ask the question whether the regeneration genes that we identified were globally differentially regulated by injury. That is, does their expression change with injury? And to put it succinctly, no, it doesn't. We were able to evaluate this because some of our co-authors recently published an RGC single-cell RNA-seq data set that allowed us to measure and evaluate changes in gene expression of our novel regeneration genes. So up on the top, we have scatter plots both before and following optic nerve crush injury, which established that the majority of our regeneration genes are expressed significantly within the RGC population. But you'll notice that the expression levels don't change appreciably with injury, which is perhaps better viewed in C here in the bottom left. So really this just showed that following injury, the gene expression levels of our regeneration genes are not altered by injury or injury signals, which does fall in line with other positive regeneration genes such as P10. We also found out that our genes are indeed quite diverse and do not have very many overlapping pathways or mechanisms of action. But what we did find is that there were 4 genes that are involved in cytokine signaling: IL-17C, IL-17 receptor B, SOX4, and IL-22. An expansion of that network identified the JAK-STAT signaling pathway and STAT3, which is really rather a canonical indicator of regeneration in both the peripheral and central nervous system following injury. So we decided to explore that pathway further and focus specifically on IL-22 because it had the most robust regenerative phenotype after editing both in vitro and in vivo. So the remainder of this talk is going to focus on that IL-22 mediated mechanism. And a hallmark of successful nerve regeneration after injury is the stimulation of a neuron's intrinsic growth capacity. And that is most often measured by looking at regeneration-associated genes, or RAGs. So we decided to evaluate whether IL-22 editing primed RGCs to regenerate. And so we measured changes in transcript levels of several RAGs, including ATF3, GAP43, SPIR1A, and galanine at 3 days post-injury in the retina. And we observed between a 2- and 4-fold increase in the transcript levels of ATF3, SPIR1A, and galanine following IL-22 editing relative to the non-targeting LAC-Z control. And moreover, we were able to determine an increase, a significant increase in the immunoreactivity of ATF3 following IL-22 editing compared to control. Now, in order to bridge the IL-22 editing regenerative phenotype that we observe and this increase in RAG expression, we decided to evaluate STAT3 expression, since it sort of brings everything together, since IL-22 is involved in JAK-STAT signaling and STAT3 has been known to induce RAG expression. And so when we look at changes in transcript levels of STAT3, we see a significant increase following IL-22 editing 3 days after injury. And that translates into increased activation of phosphorylated STAT3 14 days after injury with IL-22 editing compared to non-edited injured retinas. What perhaps is most interesting is that with IL-22 editing alone without injury is sufficient to induce this activation of STAT3, which suggests that IL-22 editing alone can partially bypass that necessity for injury to induce STAT3 activation. Now, aside from STAT3, there are other regeneration signals or injury-induced signals that lead to activation of RAG expression. And one such signal is dual-leucine kinase or dual-leucine zipper kinase or DLK. And DLK not only acts at the level of the RGCs to induce the RGC's regenerative potential, but it also can act at the level of the axon and can ferry phosphorylated STAT3 from the axon to the cell body of the RGC to induce regeneration in that manner. So we decided to evaluate DLK levels in the retina at 3 and 14 days after injury and observed quite clearly an increase in DLK protein 14 days after injury following IL-22 editing. Compare that to the Laxi unedited control. And the majority of this DLK we found to be contained within these beta-3 tubulin positive neurons that are in the ganglion cell layer where you would find those RGCs. So taken together, these results suggest that amplified expression of RAGs following IL-22 editing and inhibition is driven by both an increase in DLK and an increase in STAT3 activation. Now, to establish a connection between IL-22 suppression and STAT3 activation, we surveyed retinal expression of multiple STAT3 activating cytokines in response to IL-22 loss. And what we observe across the board 3 days after injury in the retina is an increase in the levels of these pro-inflammatory cytokines that are known activators of STAT3. Probably most significantly known the best are LIF and IL-6. Now, this suggested to us that there was an increased inflammatory response within the retinas themselves. And sure enough, we did also see an increase in the chemokine CCL2 that has been known to induce accumulation and chemotaxis of microglia and macrophages. And when we actually looked at the accumulation of cells within the retina, we observed 3 days after injury, a significant increase in IBA1 positive cells and CD68 positive cells in IL-22 edited retinas compared to control. But this increase in the accumulation was rather transient. We observed it at 3 days post-injury, and then it sort of normalized to control Laxi levels at 14 days after injury. And so what we observe is that with IL-22 editing, there is this short-lived but robust increase in inflammation by the presence of microglia and macrophages in the retina that leads to increased expression, transcriptional activation of pro-inflammatory cytokines that induce expression of STAT3 and RAG expression, and ultimately leading to increased regeneration. Now, I know that the majority of this talk was spent on IL-22 and its mechanism of regeneration, but I do want to reiterate that our regeneration screen identified more than 36 novel regeneration genes whose mechanisms are currently being explored. And so current and future studies are looking into how those genes are involved in regeneration, what's going on via various pathways, and also looking into multiplex CRISPR-Cas9 gene editing with the idea that if you target more than one gene simultaneously, you can have additive, or better yet, synergistic effects on regeneration. So those studies are currently underway. And so I'd like to say that you can access all of the in vitro cortical neuron and then the in vivo optic nerve regeneration data at the St. Jude AGI Discovery Science portal. And with that, I'd like to thank everybody who is involved in this study, all the collaborators, Steve, and I'd like to thank you all for your attention. Thanks.
Hi, my name is Konstantin Levi, and I am scientist at Kevin Park's lab in the University of Miami Miller School of Medicine. The main focus of our lab is axonal regeneration, and the title of my topic today is Identification of New Factors to Improve Retinal Ganglion Cell Survival and Regeneration, Insights from Distinct RGC Subtypes. Poor regeneration of retinal ganglion cell axons presents a major obstacle in treating of ocular trauma or disease. RGCs are very diverse cells, and some of them are capable of regeneration while others not. We set up the following aims for our study. Assess the role of genes differentially expressed in the regenerative and resilient retinal ganglion cells on promoting neurite outgrowth. Define lipidomic signatures unique to IP RGCs and test distinct lipids on neurite growth. And determine the effects of candidate genes on lipids and RGC survival in regeneration in vivo. More than 30 different subtypes of RGCs have been identified so far based on functional, morphological, and molecular features. Response to injury is one of the main features. We will focus on two types of RGCs. Intrinsically photosensitive RGCs or IP RGCs. They are characterized by expression of melanopsin encoded by OPN4 gene, resilient to cell death and highly regenerative. And we compare them to on-off direction selective RGCs. They are very sensitive to the injury, die within 2 weeks, and fail to regenerate. Here we tested regenerative ability of IP RGCs by utilizing a transgenic mouse with Cre recombinase or inducible Cre inserted into OPN4 gene. These mice were crossbred with transgenic mice expressing Cre-controlled ROSA26 TD tomato reporter. This way, OPN4 expressing cells could be revealed by TD tomato fluorescence. You can see the retina on the left side. Prior to crash, mice were injected with adeno-associated virus vector expressing CNTF, a known promoter of regeneration and neurotrophic factor. As you can see on the right, there is a section of optical nerve 6 weeks after the crash. The crash site is on the left edge. Magnified portion shows a large number of IP RGCs axon labeled with TD tomato and regeneration past the crash site. CTB labels neuronal tracer. It's a colored toxin beta subunit. High regenerative capacity of IP RGCs is not limited to CNTF treatment that activates STAT3 signaling pathway. It also happens in mice with deletion of P10 phosphatase, a known inhibitor of regeneration which relies on mTOR signaling pathway. As shown here, 3 weeks after the crash, there is a lot of regenerating IP RGCs. This number significantly increased in mice with P10 knockout. On the other hand, direction-selective ganglion cells do not regenerate even in the absence of cell death. We looked at another mouse model, HB9, expressing GFP reporter in direction-selective neurons. To prevent injured cells from cell death, we crossbred HB9 mouse with transgenic mouse lacking known apoptotic gene bugs. After the injury, cells stayed alive but would not regenerate past the lesion site. With these two mice on hand, we isolated RGCs and subjected them to RNA-seq analysis. Retinas were dissociated 3 days after the injury, and single RGC were manually collected under fluorescent microscope using micropipette. 3 pools of 10 cells were combined into 1 biological sample. Altogether, we had samples with or without crash. Clustering of analyzed data revealed small intergroup differences and distinct separation between OPN4 and HB9 groups. Differential gene expression was defined as at least twofold difference with adjusted p-value equal or below 0.05. Total 11 genes were found uniquely expressed in injured OPN4 RGCs. This volcano plot shows differential expression. We found that THBS1 is the most significantly differently expressed genes when compared to injured OPN4 and HB9 RGCs. THBS1 is a gene that encodes multidominic matrix protein thrombospondin 1 that promotes neurite outgrowth in neurons in vitro. However, the role of neuronally expressed THBS1 is unknown. Using AAV-mediated shRNA knockdowns of THBS1, along with CNTF stimulation 3 weeks before crash, we show that thrombospondin 1 is indispensable for regeneration. Overexpression of thrombospondin 1 promotes spontaneous regeneration with many axons growing more than 1 millimeter beyond crash site. Since thrombospondin 1 is a large protein with multiple functional domains in its structure, we created a series of truncated mutants and expressed them in retina. We found out that TSR1 domain is not required to promote regeneration in vitro. However, CTD and coiled domains are necessary. These domains also facilitate the trimerization of protein. We also compared this data to thrombospondin 4, which is not expressed in RGCs and does not promote regeneration. Collectively, this data show that CTD and coiled domain and trimerization of the protein are required for regeneration of axons in vivo. Next, we return to our data obtained in RNA-seq in order to examine other potential positive regulators of regeneration. We used a list of upregulated genes and reselected them based on the following criteria. They should not be expressed in HB9, expressed but not changed in HB9, expressed by OPN4 and downregulated in HB9 after the optic nerve crash. They could be expressed and upregulated in HB9, but OPN4 expression after the crash should be at least 5-fold higher than HB9 after the crash. This selection revealed about 400 genes meet the criteria. Next, to test the selected genes, we set up in vitro screen using siRNA pools and rat hippocampal neurons. Neurons were isolated from E18 hippocampi and seeded in laminin-coated plates. Next day, a cell siRNA pools were used for treatment. And 4 days later, cells were fixed, stained with beta-tubulin neuronal marker, and the length of growing axons calculated using Celomics array scan and automated tracing software. We screened a total over 160 genes. And after the initial screening, 24 genes were selected for further testing. After the data were normalized, 4 genes were selected for individual siRNA analysis. This is needed because siRNA pools contain a mixture of 4 different siRNAs to the same gene, and each one have to be retested separately. One of the genes, RPH3A, became a focus of our further investigation. RPH3A gene encodes rapfilin 3A protein that contains a calcium-dependent membrane-targeting domains. It was shown earlier to regulate neurotransmitter release in hippocampal neurons. And it is known, its knockdown consistently inhibited neurite growth in our in vitro experiments. To reveal its potential role in axonal regeneration, we decided to specifically suppress it in RGC. We generated AAV vector for CRISPR-Cas9 gene editing in RGCs. Recently, Elizabeth Simpson and colleagues in Canada defined a minimal promoter from neurofilament light chain gene with specificity for ganglion layer cells. In our constructed vector, this promoter will ensure tissue-specific expression of Cas9 in Cre-dependent Cas9 transgenic mice and enable targeted editing by CRISPR. You can see in the picture that transduction of retinas with this vector cause the expression of Cas9 in ganglion layer cells selectively, along with GFP reporter. Design and validation of single guide RNA for CRISPR-Cas9 mediated knockdown was performed using algorithm Cas5 maintained by Hanyang University in Korea. Selected as small guide RNAs tested and validated in vitro using mouse neuronal 2A cells and surveyor nuclease mutation detection kit. We also tested and designed and validated small guide RNAs for CRISPR-Cas9 mediated knockdown of some transcriptional regulators highly induced in direction-sensitive ganglion cells after the injury. This is ongoing work, and we still don't have any results yet to show. Our future goals. Use AAV CRISPR-Cas9 shRNA to continue and examine the roles of differentially expressed genes identified in our studies in promoting RGC survival and axon regeneration. Examine whether these molecules promote regeneration in the wide variety of RGC subtypes. Examine whether regenerated axons find brain targets and lead to functional recovery after the injury. And in the end, I would like to acknowledge all our collaborators from the University of Miami participating in this research, especially Eric Bray and Benjamin Younger, who spearheaded the thrombospondin 1 studies, and our collaborator from Northwestern University, Tiffany Schmidt, who provided us with OPN4 Cre transgenic mice. And we thank National Eye Institute for the support of this study. Thank you.
First of all, I would like to thank to the organizer for opportunity for me to share our work here, and thank you everyone for the time. I would like to share with you the work that we have done on the retinal regeneration from nuclear cells. I have no financial interest to disclose. As you probably know that vision loss is among top 10 disability in US. Million people got affected by retinal degeneration, including diabetic retinopathy, glaucoma, and AMD. It is estimated that the number will double by 2050 with increasing in aging population. There are several potential therapy approaches for retinal degeneration, include gene therapy for treating some of the blinding disease where the cell is still viable. Otherwise, cell-based therapy will be needed. This includes transplantation of retinal neuron or reprogramming the resident nuclear cell in the retina. In the retina, nuclear cells have radiomorphology and expand through different layers of retina. They provide structural and functional support for the retina. During development, nuclear cells are among the last born cell type. Our lab recently performed single-cell ataxic analysis of retina during development and showing that individual development trajectory of individual cell type, including retinal ganglion cells, amacrine and hotel cells, cold and rod cells, macular cells. Nuclear cells are formed from late-stage protein cells and transcriptionally resemble late-stage protein cells. Significant effort has been made to reprogram nuclear cells for treating retinal degeneration. Inhibition of people pathway result in nuclear cell proliferation in mice. However, they do not efficiently produce retinal neuron. In other studies, overexpression of KR4 and NEOZ2 result in limited conversion of nuclear cell to retinal ganglion cell-like cells in vitro. In recent landmark study by Tom Ray Group, they show that overexpression of ASCO1 with combination of histone deacetylase inhibitor and retinal injury, molecular cell can be directly converted to bipolar and amacrine cell-like. All of these indicate that we need to have a more complete understanding of genetics and epigenetics barrier of retinal molecular cells reprogrammed in mammal. Just like any other tissue in the body, the ability of retina to regenerate has been lost during the evolution. In zebrafish, for example, injury can lead to retinal regeneration throughout life. If we depleting the photoreceptor layers, molecular cells will undergo proliferation and migration to the photoreceptor layers, where they can give rise to the photoreceptor layers and rescue the visual function. In chicken, the regeneration only happens transiently in early life, 2 weeks after hatching, and only limited number of neurons can be produced from molecular cells. In contrast, mammals, including human, injury can only lead to reactive gliosis but no regeneration. So we want to ask how molecular cells respond to injury differently across different species, and what we can learn from fish and chick to reprogram mammalian molecular cells. So we decided to perform cross-species transcriptomic and epigenomic analysis of molecular cells in response to different stimulation. We reasoned that using different stimulation, we can find the core mechanism that underlie molecular cell reprogramming. This includes NMDA treatment, which kills amacrine and ganglion cells. Light damage kills rod and cone. Growth factor treatment can lead to molecular cell reprogramming independent of retinal cell death. We also perform molecular cell analysis during development across 3 different species. After that, we perform single-cell RNA-seq analysis on the whole retina. When the transgenic animals are available, we perform FACS sorting for molecular cells and provide both RNA and ataxic analysis on molecular cell enriched and molecular cell depleted fraction. After that, we integrate RNA-seq and ataxic and build gene regulatory network and find potential key regulator. Subsequently, we perform gain and loss of function to check for their potential role in molecular cell reprogramming. By looking at both RNA-seq analysis, we can see clear difference in molecular cell response to injury among species. In mouse, both NMDA and light damage, molecular cells show rapid transcriptional change after injury, and then they reverted back to a state that closely resembles the resting state. In contrast, in zebrafish, both light damage and NMDA, molecular cells show rapid transcriptional change, and then diverted from the resting state, consistent with notion that they undergo reprogramming and give rise to neuron. We next perform single-cell analysis on whole retina after injury and combine multiple samples together. In all 3 species, we can identify all major retinal cell types, and we also identify activated and resting molecular cells. We next subset the molecular cell population and perform a pseudotype analysis. Consistent with both RNA-seq, we showing that molecular cells undergo rapid transcriptional change after injury. However, they reverted back to the states that closely resemble resting state. In contrast, in zebrafish, 4 to 10 hours after injury, molecular cells occupy the gliotic branch. From that time point, they undergo neurogenesis and proliferation. In chicken, 24 hours after injury, most molecular cells occupy the gliotic branch. From that time point, some of them undergo proliferation, and most of them come back to the resting state. We next integrate all RNA and ataxic together and build gene regulatory network to control molecular cell activation. We primarily focus on transcriptor because they are directly regulate gene expression and underlying biological process. In zebrafish, we identified 97 transcription factors and grouped them in 10 different modules. These modules cross-activate and repress each other. Likewise, in the mouse, we identified 156 transcription factors and grouped them in 10 different modules. We then draw a simplified regulatory modules of molecular cell activation. In zebrafish, we observe two cross-repressed modules, resting and gliosis. Once a molecular cell enters the gliosis state, they will progressively undergo neogenic and proliferation. Genes involved in gliosis state including HM1GA1, SMARCA5, and YAP1. Similarly, in the mouse, we observe two cross-repressive modules, resting and gliosis. However, the presence of the third module we call restore-rest will repress the gliosis state and inhibit the nuclear cell reprogramming in the mouse. The strong candidate gene for resting and restore-rest are NFI-ABX factors. We next test the effect of inhibition of key candidate genes that are activated during the gliosis on nuclear cell reprogramming. We found that knockdown either HM1GA1 or YAP1 lead to significant reduction of nuclear cell proliferation. We also treat retina with protoporphyrin, which is YAP1 inhibitor, and we found that YAP1 inhibitor lead to reduction of nuclear cell proliferation in those different manner. To test the effect on retinal regeneration, we examine retina 10 days after injury with rod and cone marker. We found that knockdown of either HM1GA1 or YAP1 lead to significant reduction of rod and cone 10 days after injury. In the mouse, since NFI factors are predicted to maintain and restore quiescence of nuclear cells, we perform depletion of NFI factor ABX in mouse nuclear cell through conditional deletion. After 21 days after injury, we perform single cell analysis of the nuclear cell positive fraction. You can see that NFI knockout nuclear cell form clearly distinct cluster compatibility control. Through clustering analysis, we found that nuclear cell division in NFI give rise to different cell types, including neogenic nuclear cells, bipolar cell-like, and American cell-like, and also proliferative nuclear cells. Using virus for lineage tracing, we found that nuclear cell-derived neurons have bipolar cell-like morphology and American cell-like morphology. And using one marker for bipolar cell type, SCGN, we found that nuclear cell-derived neuron send process to OPL and INL layer. In summary, our data identify gene regulatory networks that control nuclear cell quiescence, gliosis, and neogenesis among 3 species. After injury, mouse nuclear cell arrested in a gliotic state, while chicken and zebrafish nuclear cell transition to gliosis to neogenic state. And we show that genes that activate during the gliotic state are required for nuclear cell reprogramming in chicken and zebrafish. And finally, we're showing that selective depletion of NFI factor, which required for nuclear cell quiescence in the mouse, enable nuclear cells to reprogram and regenerate neuron after injury. All the data that generated through this project has been uploaded to St. Jude server. If you follow this link, you see all the data set in different panels. If you click in one of the panels, for example, retina following NMDA injury in mouse, that will lead you to single cell data set from mouse NMDA injury. And you can see different cell cluster, and you also can plot different gene expression. Finally, I would like to thank all people involved in this work, all members of SEP lecture lab, Dr. Jiang lab in JSU, who involved in bioinformatics analysis, David High lab in Notre Dame for zebrafish work, Andy Fisher lab in OSU for chicken work, and John Ash lab in University of Florida. This grant has been supported by NEI and Audacious Go Initiative grant, and I'm happy to take any question.
Good afternoon. My name is Jim Patton from Vanderbilt University. No conflicts to report. I'd like to thank the organizers for giving the chance to talk, and just want to give a shout out to the people who did the work and our collaborators, Ed Levine and David Calkins, and support from the Institute and the Audacious Goals Initiative. So today I'm going to tell you a story about extracellular vesicles and retina regeneration, and it stems from work in my lab that started several years ago, where we started to uncover the role of microRNAs at different stages of retina regeneration. So as many of you probably are aware, constant intense light damage and other means of damage in zebrafish initiates or induces a spontaneous form of retina regeneration that is led by Mueller glia, as first showed by Pam Raymond, that undergo a dedifferentiation event, asymmetric division, and then this stem cell undergoes multiple rounds of, or neuroprogenitor cell undergoes multiple rounds of division to replace any lost cell type. And in some work shown down here below, this is the Mueller glia. This is from Ryan Thummel from back in David Hyde's lab, and shows very nicely the Mueller glia. And then the red is the proliferating cells derived from Mueller glia that are expressing proliferating cell nuclear antigen. And I'll be using a similar tools as we go through here. So my lab, led by Kamir Rajaram, originally purified RNA at all these different stages, and have been working on identifying microRNAs that go up and down during expression, and then identifying the genes that they target. We've published on MIR203 PAC6 and MIR216 and .1L. But today I want to tell you a different story. And it stems from work in another part of my lab that also looks at RNA research. And in that case, it's extracellular vesicles in RNA. So it's now known that extracellular vesicles are secreted by all cell types and that they can play a role in cell-cell communication. Best examples are in the immune system, but it's now shown that you can get cargo transfer, and we've shown that you can get microRNA transfer from a donor to a recipient cell. This has led to all kinds of possibilities on altering the metabolism of recipient cells and for therapeutic applications. So the reason we jumped into this in terms of approaching it from the regeneration standpoint in the retina goes from work that Bob Coffey's lab here at Vanderbilt in collaboration, well, we now collaborate with Bob, but before that time, Bob had shown that extracellular vesicles from mutant KRAS cells, these DKO1 cells, when exposed to wild type cells, could cause the wild type cells to become invasive into matrigel and proliferative. Okay, so the extracellular vesicles from mutant KRAS cells were inducing these effects without inducing any genetic change. So cargo transfer from the mutant KRAS cell to the wild type KRAS cell caused the wild type cell to start to look and act like a cancer cell. So we thought to stimulate retina regeneration, would this also work? Could we induce a Mueller glia derived proliferative response by injecting extracellular vesicles? And support for that, when we put the grant together, stemmed from observations from Rachel Pearson that some of the transplantation experiments, which had previously been thought to stem cells being integrating into the retina, perhaps what was actually happening is that some of these stem cells are releasing what we would call extracellular vesicles, they called material transfer, and contents from the stem cells are being taken up by host cells, okay? So there was some support that there could be vesicle cell-cell communication. So we set out to do a large-scale screen, and we're lucky enough to get a grant from the Audacious Goal Initiative, David Calkins, Ed Levine, and I. And the plan was to purify extracellular vesicles from many, many sources, inject them intravitreally into fish, and monitor those that would induce Mueller glia-derived proliferation. First, it would be done in fish, and then shortly thereafter and somewhat concurrently into mice, and that Ed Levine's lab would take the lead in then deciphering whether these extracellular vesicles were just leading to proliferation, neurogenic potential, or photoreceptor cell fate. Today, I'm only going to tell you about the fish story. Ed is working hard on the mouse aspects of this. So this was an in vivo screen, kind of a risky and difficult screen. And to make a long story short, these are three of the best cell lines that we did this. Okay, so here is the results of the screen, or initial screen, ongoing screen. So 59 different extracellular vesicle preparations were intravitreally injected into an otherwise undamaged fish. 72 hours later, we monitored DNA replication by pCNA staining, immunostaining. And shown here is that compared to the PBS control, many of these extracellular vesicles didn't do anything, but the ones over on the right gave a response. I need to point out that this response is not at all as robust as you would see after damage, but we did not damage these eyes other than injections. So the best of these was this C6 glioma shown over on the right, and that's primarily what I'll work forward. So the initial screen was done with a somewhat crude pellet of extracellular vesicles. So there are ways to purify these vesicles. I'll show you in another slide, but extracellular vesicles come in a variety of sizes, and they are through different biogenesis pathways. The most well-known of these are exosomes from the endocytic pathway, but all cells released both from the plasma membrane and via these multivesicular bodies, a heterogeneous group of vesicles. Our control experiments suggested that the major activity was in these small extracellular vesicles, which were traditional exosomes, although we did see some activity in the larger microvesicles. So to refine that better, we utilized purification techniques that were just recently revised by Dennis Jeppesen and Bob Coffey's lab. So instead of our initial pellet that I showed you on the initial screen, then we further purified the extracellular vesicles and came up with these pools and tested the pools, and pool 2 gave the most regeneration, or the most proliferation. I should always be careful how I phrase that. And pool 2 consists of what are traditionally referred to as exosomes based on these markers that are commonly found in exosomes. Okay, so once we had the really more highly purified vesicles that we knew were giving a response, we conducted small RNA-seq and proteomics here at Vanderbilt Cores, and all of that data has been submitted to the St. Jude's portal as part of the Audacious Gold Initiative, and I welcome everyone to mine it. We are mining it ourselves. Okay, now to give you a little better flavor of that this was actually working, here's an experiment where we injected the extracellular vesicles from the C6 cells, and then we utilized morpholinos against the transcription factor ASCL1. So most of you probably know that ASCL1 is required for retina regeneration. So we knocked down ASCL1, and shown here, so this is injection of the C6 extracellular vesicles in a control morpholino, and then the C6 extracellular vesicles with knockdown of ASCL1, and you can see an incredible decrease in proliferation. These experiments were using a line of zebrafish that labels the dedifferentiated Mueller glia, and so you can see that this is sort of following, not sort of, it's following a more well-known bona fide regenerative cascade. Okay, this is the extent of proliferation we were getting just by injecting a standard control morpholino. This is with the C6, so you can see the effects of the C6 extracellular vesicles pretty easily here. Okay, this top 4 panels were done with the more crude pellets. This one down here is with the purified pool 2 vesicles in exosomes, and this is again using the tuba line. So we have extracellular vesicles produced by the C6 glioma line that turned out to be the best source of extracellular vesicles for inducing Mueller glia-derived proliferation in the zebrafish. So that begs the question, how can we increase the effect of these extracellular vesicles? I told you that it's not as robust as damage, and indeed we would like to get many, many more Mueller glia to re-enter the cell cycle, and we would really like to be able to do this in an undamaged setting in a way to trick Mueller glia into think there is damage. So right now, the use of exosomes and extracellular vesicles as therapeutic tools is a burgeoning field with many companies involved. The idea here is that you can load these extracellular vesicles up with cargo proteins or small RNAs. You can attach or incorporate membrane proteins that could target them to specific cells. We would like to increase the concentration of regenerative factors, proteins, RNAs inside, and we would like to have receptors or ligands or antibodies that will target these vesicles to Mueller glia and induce the response. Our first foray to do this, we just took our C6 cells and then we overexpressed 4 factors that are known to induce proliferation, Mueller glia-derived proliferation and regeneration. And when we did that, overexpression of IL-6 and overexpression of ASCL1 both induced a more significant response than the parental C6 EVs. So this gives us hope that there is a chance where we can create designer EVs to stimulate retina regeneration. Okay, so conclusions: Mueller glia in the fish serve as adult stem cells in vertebrate retinas. Spontaneous retina regeneration in the fish is blocked in mammals, and we would like to be able to find ways to induce a regenerative response in mammals. Using extracellular vesicles, we've shown that we can, in some ways, deliver cargo. We don't know exactly the cargo. That's ongoing work, but it shows that you can, from other stories and from our own, load these vesicles up with mRNAs, microRNAs, proteins, small molecules, and then there are tools right now to target surface molecules on Mueller glia to increase the uptake of these EVs. So with that, I'd like to conclude. Thank you, and shout out support to the National Eye Institute and the Audacious Goals Initiative. Thank you very much.
Back to basics. Informing repair strategies by first understanding normal photoreceptor axon growth. I, Sarah Rempel, have no conflicts of interest to disclose, and neither does Timothy Gomez, Cindy Berlinick, or Don Zach. David Gam has a personal financial interest in, is a consultant for, and holds patents with Optic Therapeutics. Photoreceptors are the primary sensory cell that allow for image formation and perception of vision. This means that when photoreceptors are lost due to disease or damage, it causes blindness. So if the problem is that photoreceptors are gone, could we cure this blindness by just transplanting new photoreceptors in? In order to have that happen, we don't just need the photoreceptors to be present in the retina, we need them to form synaptic connections with the host bipolar cells to integrate into that existing circuit. And in order to form those new synapses, donor photoreceptor axon terminals will need to be positioned next to bipolar cell dendrites, presumably through axon extension and guidance. This process of axon growth and guidance has been well characterized in projection neurons like retinal ganglion cells, spinal cord motor and sensory neurons, or hippocampal and cortical neurons. A newly born neuron is roughly spherical and then must polarize. At the tip of the growing axon is a structure called a growth cone that autonomously navigates the environment to reach its target, where it elaborates a synaptic terminal. This movie from Gomez Lab alumnus Timothy Catlett shows the growth cone of a human stem cell-derived cortical neuron, and you can see that these are dynamic structures that exert forces on the substrate to extend out the axon behind them. With this problem of photoreceptor transplant and our knowledge of growth cone-mediated axon extension in mind, we developed our AGI grant proposal to, firstly, identify small molecules that promote the axonal outgrowth and guidance of human stem cell-derived photoreceptors. Secondly, identify novel molecules that enhance human stem cell-derived photoreceptor synaptic marker expression. And thirdly, develop secondary assays to assess photoreceptor process extension and functional synapse formation in culture, and use these assays to test the activity of lead molecules identified in AIMS 1 and 2. For this talk, I'll be focusing specifically on our findings related to photoreceptor axon growth and guidance. The Zach lab, including Don, Cindy, and Bibhu, were our high-content screen experts, having successfully used the approach to identify factors that promoted axon extension of retinal ganglion cells. The Zhao lab, including Zhenyu and Yu, provided their expertise in synaptogenesis and synaptic circuit tracing for that second aim. The GAM lab, including David, Joe, and Stephen, provided their expertise in stem cell-derived retinal organoids and photoreceptor development. The Gomez lab, including Tim, my undergrad Maddie, and me, were there to provide our expertise in growth cone dynamics and axon outgrowth and guidance. The GAM lab generated retinal organoids from human stem cells, specifically from a line in which all postmitotic photoreceptors express TD tomato driven by the native CRX promoter. They then sent these retinal organoids to the Zach lab. Preliminary imaging done by Joe in the GAM lab had found that photoreceptor axons seemed to be longest around day 80, so we chose that as the time point for treatment. So at day 80, the Zach lab dissociated the retinal organoids and treated with a panel of kinase inhibitors. These plates were then imaged and automatically analyzed using that TD tomato fluorescence signal to measure photoreceptor neurite length. From that kinase inhibitor screen, they identified a few drugs that caused increased photoreceptor neurite length. The x-axis is just the index of each molecule in the screen, while the y-axis shows the mean photoreceptor neurite length represented as the percent of the control photoreceptor neurite length. These hits that all consistently increased neurite length all had something interesting in common. They all had ROCK inhibitor activity. ROCK, or Rho-associated protein kinase, is a downstream effector of a Rho-A GTPase, one of the master regulators of the actin cytoskeleton. Its kinase activity promotes actin polymerization and stabilization through limb kinase and cofilin, and its phosphorylation of myosin light chain promotes actin contractility. Therefore, inhibition of ROCK leads to decreased actin contractility and increased actin turnover, resulting in faster extension. And this faster growth has been seen both in projection neuron axon extension rates, but also generally in migration and motility of a variety of cell types. This is where I came in, to validate the hits with small-scale experiments. So I repeated the process of differentiating retinal organoids, again using this photoreceptor reporter line, then dissociating the organoids into a 2D culture. I then treated these cultures for 24 hours with 2 of the ROCK inhibitors from the screen. I then imaged individual photoreceptors live for 20 minutes with confocal microscopy. I was surprised to see no difference in growth cone displacement of photoreceptors treated with the drugs compared to the DMSO control, with hardly any displacement greater than 1 micron over the 20-minute imaging period. Additionally, for photoreceptors sitting alone, not contacting any other cells, they showed virtually no axon length change with either of the drugs. However, among photoreceptors that were touching another cell during the imaging period, I did see greater length change in those that were treated with one of the 2 compounds. And as we continued to explore the growth characteristics of these human stem cell-derived photoreceptors, it became clear that photoreceptors had much longer neurites when contacting another cell in the dish than those photoreceptors that were sitting by themselves. And these growth cone-looking terminals didn't actually seem to be capable of growing at all. As you can see in these movies, these photoreceptor terminals seem to stay stuck, immobile to the substrate. And photoreceptors not contacting other cells have a mean neurite length of only about 20 microns and are virtually never longer than 50 microns, while those that are touching other cells have mean lengths closer to 40 microns with many over 50 microns. Longer-term live imaging made it clear that these axons didn't result from autonomous growth cone-mediated growth. You can see in these images that between 1 to 2 days in vitro, the cell body of the photoreceptor at the bottom, highlighted with blue arrows, actually gets pulled away from where it had previously been by a cell that wasn't present at 1 day in vitro, leaving a stuck down terminal behind. Then at 3 and 4 days in vitro, that bottom cell body actually returns to its former location while the upper cell, labeled with yellow arrows, undergoes a similar axon extension due to cell body movement. So it appears that this photoreceptor extension is not happening because of any autonomous growth, but because other cells in these heterogeneous cultures are pulling the cell bodies of the photoreceptors away from those adherent terminals. So this recontextualized all of our high-content screen results. ROCK inhibitors likely influenced motility of other cells in the dish and weren't affecting the photoreceptor growth cones directly. That high-content screen approach had worked really well for retinal ganglion cells, so why were our assumptions so wrong for photoreceptors? While retinal ganglion cell neurite growth and growth cone dynamics have been extensively studied for decades, it turns out we just know incredibly little about mammalian photoreceptor axon extension and development. In the early 90s, Cleavin and Ray cultured rat rods on a variety of substrates or co-cultured them with Mueller glia, and found that co-culture with Mueller glia caused the rods to have much longer neurites. Then in the late 90s, Johnson and the Reese lab found that in ferret, rods overproject to the inner plexiform layer early in development before retracting back to the outer plexiform layer. And Hendrickson and Zhang reported this overprojection to the inner plexiform layer in cones in human fetal retina in 2017. But that handful of papers spread over the past 30 years represents the majority of what we knew about mammalian photoreceptor axon growth prior to starting this project. So I'm going to end by telling you a bit of what we found about the axon extension and growth cone dynamics of human stem cell-derived photoreceptors in work that has sprung from this AGI grant. As you saw before, at 80 days of development, photoreceptors don't seem to make dynamic growth cones, but instead have these stationary terminals. I started exploring different ages, however, and found that at day 40, photoreceptors often had dynamic growth cones like we see in other types of neurons. My undergrad Maddie and I went through each movie blinded and categorized photoreceptors as having dynamic or stationary terminals, then averaged the percent that were dynamic in each dish by age, and saw a clear reduction in the percent of photoreceptors with dynamic terminals with age from about 70% dynamic at day 40 to about 5% dynamic by day 80. Now, I've previously been lumping all photoreceptors together, but of course there are actually two major types, cones and rods. And they're born at slightly different times during development, with cones being born first, starting around day 30 in these human retinal organoids, and rods being born second, starting around day 70. So I wondered whether this decrease in dynamic photoreceptors could be a result of a difference between rods and cones. To address that question, I used a new stem cell reporter line, this time with GFP driven by the rod-specific transcription factor, NRL, so that all of my rods fluoresce green. I dissociated and imaged them as before. Out of over 600 rods across day 80 to day 127, I never saw a rod with a dynamic growth cone. Therefore, it seems that while human stem cell-derived cones, at least at younger ages, can make the dynamic growth cones that are necessary for autonomously extending their axons, rods are never able to do so. So what does that mean for photoreceptor transplant? It means that we can't necessarily rely on transplanted stem cell-derived photoreceptors to do the heavy lifting of extending an axon to the appropriate target. We'll either have to identify the regulatory factors that control this loss of dynamicity and manipulate photoreceptors to return to that dynamic state, or we could focus on strategies to encourage bipolar cell dendrites to extend to meet the transplanted photoreceptors where they are. Either way, we'll clearly need a better understanding of the development of photoreceptor axons to realize the promise of transplant therapies to cure blindness. In terms of the AGI grant, we also learned that while high-content screens work well in systems where we already have a lot of background knowledge, that approach may be more limited in situations where that basic research just isn't there yet. With this context and awareness that our understanding about photoreceptor neurite growth is still limited, the data from our neurite length screen is up on the St. Jude portal, where you can access it. Also, this area is still wide open, and I hope my talk has gotten some of you curious and excited to research some of the many remaining questions we have about how photoreceptor axons get to the outer plexiform layer during normal human development. Thank you for your attention. Thank you to ARVO for hosting this symposium, and thank you to the National Eye Institute for funding this research.
Well, hello, I'm Michael Chang, and I'm the new director of the National Eye Institute and the live portion of this symposium. So thanks to all of you for joining this ARVO special session that really features our AGI consortium projects about discovering new regenerative medicine factors. So just to say up front, if you have questions, please enter them into the chat box. This was a really impressive set of projects that's moving us toward that long-term goal of restoring vision in people who've lost eyesight. And in this symposium, I think what we've seen is really very important first steps toward advancing toward the development of these treatments by really looking at these new factors and the approaches that are being used to find them. Of course, I want to emphasize that it's going to take more than just this group of AGI investigators to translate that work. And so I hope, and we at NEI hope that this symposium, really what it does is that it can inspire everybody in the audience to look at the function and the mechanisms of the factors that were discussed here, along with other related factors that you all know about from your own work. So the only last thing that I want to do is just give a quick shout out to the St. Jude data portal that Mike Dyer highlighted in his first talk. I'm a data person by background, and I really believe that data sharing is how we're going to advance together as a scientific community. And so, please check that out. Many of you know an African proverb that says, if you want to go fast, go alone, but if you want to go far, go together. And I think that we've all seen in this pandemic with things like vaccine development that I think the Africans got it right, and that we've really come actually both far and fast through data sharing. And so you'll hear more about data sharing through the NEI and the NIH in the coming year or two. But in some, please consider Mike's data portal in your own work. So with that, Mike, I'm going to turn it over to you, and thank you for moderating this discussion. Steve Becker, thank you for helping to organize it all.
Perfect. Thank you so much. It's been a real pleasure serving on the oversight committee for this group. And I think, the NEI has really been pioneering in the sense of taking on these high risk, high reward projects. And you've seen from the talks just the incredible impact that has had. And our goal is to put the data in the hands of the researchers as quickly and efficiently as possible. So there are a lot of questions, and I've started to type them up and collate them. So I'm just going to go through each one of the speakers and give you a question, and then we'll keep going until we run out of time. So we're going to go ahead and, there was two quick questions on the cloud, but I'll just quickly address those. The first was about network analysis. And the real vision for this is twofold. If you're somebody like me who's not a computational expert and you just want to mine the data, we made visualization platforms that allow you to do that. If you're a computational expert and you want to dig into the data, you can also go and get access to the data. And the real vision is you can bring your tools to the data or bring the data to your tools either way. So it's really nimble, but it doesn't have existing network tools there. And then a question about mapping to the current mouse genome reference, MM10. That's in on our to-do list, but as you know, these things take a bit of time. So we're going to try to get that down the road. All right, so my first question is for Sahil. Really impressive, beautiful work on KIF5A. And you made the point that when you tried to ectopically express KIF5A to rescue the loss of function, it didn't rescue, and in some cases, it actually made it a little bit worse. I thought that was really fascinating observation. So I thought maybe you could comment a bit on that. And do you think there's some sort of compensation when KIF5A is deleted? And then when you try to rescue, that compensatory mechanism is preventing the rescue, or do you have a sense that something else is going on? So Sahil.
Yeah, definitely. And that was actually one of the most interesting points that we found when we tried to do this kind of standard overexpression experiment. And it makes sense when we look at our first Western blog where we showed that KIF5A expression is nearly not affected. So we think that KIF5A as a protein is being made in the cell body, but there is a regulatory element that's preventing it from moving into the axonal compartment. And if that's true, that means if we overexpress KIF5A, we're just keep on making more of it, but really not getting it out into the axonal compartment, causing more of a traffic jam on these axons. So I think that does highlight one of the interesting things that's happening in this type of injury where we may not have been monitoring what's happening in the transport section besides the RNA with transcriptome or with protein synthesis with the proteomes, but actually are these proteins functioning in the location they're supposed to? So our goal is to then, besides KIF5A and other proteins too, but figure out a way of changing the regulation to get these motor proteins like KIF5A to transport into the axonal compartment from the cell body in face of some of these stress type injuries, which includes glaucoma, but also optic nerve injury.
Incredible. Yeah, that study really convinced me of how important it is to think not just of a cell expressing a protein or a gene, but also where it is and how it's getting there, right? So really elegant work. Okay, next question is for Jane. It came up, the question is, did you call up IL-22 receptor as a hit? And what about PTEN itself, PI3K, pathway genes, the sorts of things you might expect from the screening and other genes that control, that are sort of brakes on axon regeneration, like no-go?
Sure. So the usefulness and the effectiveness of the genome-wide in vitro cortical neuron axonal regeneration screen was that it validated previously identified regeneration genes, such as P10. So P10 was definitely a hit in that screen. SOX3, others, and of course with inhibition of P10, you see an increase in the expression of PI3 kinase and AKT pathway mediators. Now, in terms of IL-22 receptor A1, it was not a hit in the 400 gene assay. The 400 novel genes that we identified in the in vitro screen. And so it wasn't something that we initially evaluated in vivo using shRNA or CRISPR AAV. But when we then identified IL-22 as one of those really potent regulators of axonal regeneration, we went back to its receptors, IL-22 receptor A1 and IL-10 receptor B, and generated guide RNA AAVs against those receptors and evaluated them in vivo. Now, with respect to IL-22 receptor A1, we didn't see an effect on optic nerve regeneration in vivo. And that wasn't entirely surprising because the RGC single-cell RNA-seq data did not find levels of expression, detectable levels of expression of IL-22 receptor A1 in those RGC populations. And now in vivo, our AAV targets really rather specifically those RGC populations. And so what we think could perhaps be happening is that IL-22 expression in RGCs is acting in a paracrine fashion on perhaps immune cells or other cells that have an influence on inflammation and the expression of other immune regulators, thereby changing those downstream expression levels of those inflammatory mediators and growth factors.
That's really exciting. I was super excited to see these pathways coming up, and I wondered, right, is it cell intrinsic and signaling to the immune system and back and forth? And I think it's really opening up a whole new area of research, right? Digging deeper into the immune system, just as we've learned in the cancer field, right? The immune system is so important for so many of these diseases, and now you have the tools to study it. Really impressive. All right, Constantine. Let's see, there was a technical question about single-cell RNA-seq you addressed in the chat, but I'll let you touch base on that. I guess another question that's come up, and it's just such an impressive axon regeneration phenotype, right? The response is remarkable. Yes. And the structure function analysis of thrombosponin 1, where you took the different domains and kind of deleted those, again, really, really impressive. And that speaks to how robust, your assay is, just to be able to do that mechanistic study. So the question is the response in this sort of an assay, is it all or nothing? So that is, if you get a response, basically you get the full length, or is it somehow graded? So you might delete a domain and get a partial response, and then you delete another domain and get another one. So do you have a sense of that, or is it not sensitive enough to pick that up?
We tested several domain deletions, and we just were looking for either positive or negative result. So we did not look at partial result.
Yeah, and it's still so impressive. So I was just sort of curious if you could tease that out further. And do you want to comment on how difficult it is to do these single cell RNA-seq analyses of ganglion cells in particular, right? Where you might get some background and specificity is really difficult to tease out.
We are very much interested in doing this single cell or single nuclei analysis. And our main object of interest is optic nerve. But preparing the reliably single cells or single nuclei from optic nerve presents a tremendous problem due to a dura matter and the presence of a large number of the fibers in the prep. And each fiber is separately tagged during the labeling of the cell or nuclei, and presents problems. So we tried several attempts So far, we did not find a way to get rid of this problem, but we keep working on that.
Right.
We cannot offer any recommendations yet.
I'll just chime in, for those trainees in the audience or those who haven't done these types of analyses, it is not trivial. It takes quite a bit of optimization, and the data are much more shallow than a bulk RNA-seq. So having some orthogonal way to analyze, or looking at other data sets, right? So that's also where,
Yes.
A common repository is very useful to cross-compare your data to other people's data. It can be really beneficial.
We had some results with like 12 or 15 cells, but
right. Sometimes you just need more. All right. So for Thon, on the beautiful regeneration story, I mean, just remarkable comparison across species. Lots of questions. I'm going to start with a developmental question, since I'm a developmental biologist, and I thought this was a really insightful question. The question is, in the zebrafish single cell RNA-seq time course of regeneration, okay, so you got your Muellers and they're regenerating, is there a point at which they stop looking like regenerating Muellers and start looking like the developing normal cells, that you can't distinguish them from just a normal developing amacrine cell or bipolar cell or something like that? Can you distinguish that?
Okay, so I have to say that, so for our study we published, we are mostly looking at from like 0 time point, like to where they undergo proliferation and neogenic state. So they haven't differentiated into different cell type yet.
Right.
Okay, but we can do like initial comparison between the developmental nuclear cells and the reprogrammed nuclear cells, and we found that the majority of gene is shared, at least to the postnatal state. Yes, and we're looking at more like difference instead later on by chasing them longer after injury, see how different injury can make the nuclear cell bias to different population cell type later on. Yes.
That's really impressive. I wonder, has anybody in the fish ever looked much later after regeneration and purified out the new neurons and asked even, are they really exactly the same as a neuron that was made during development, or is there still something maybe a little bit different about those that have come from regeneration?
Yeah.
But maybe people haven't done it.
I think that's a very interesting question, because I think I actually through this talk, I know that several groups try to do that. And normally for the retina, after injury, they fully recover after maybe 3 weeks to 1 month after injury. And depending what type of injury, there are several reports showing that they actually produce an ectopic cell type as well, which normally doesn't get killed by the injury. Yeah. So this is how they actually reform the connection to the circuitry.
Fantastic. Beautiful, beautiful work. All right, James, I've learned a lot about vesicles, extracellular vesicles from participating in this project, and it's a rapidly changing field. I guess one of the questions that's very specific to this project was, how does, proliferation response kinetics give you some clues about how the vesicles are acting on Mueller glia in particular, maybe in the fish system? So can you sort of comment on that proliferation kinetics and the exact mechanisms of some of these vesicles?
Right. Well, great question. And, I don't have a hard answer on that. And it's a little hard to do the kinetics because you're dealing with delivery of vesicles that could be going to many cells. And so lots of things could be going on. But I think at least what we're focusing on a little bit is there seems to be a requirement for also an inflammatory response. And we're not sure that we're getting that really at all with these EVs. And that's interesting in itself. So I think the MUM, Ray, and Stenkamp labs have now shown that you need to have some involvement for an inflammatory response, and it needs to somewhat resolve itself as well. So that's a injury dependent. Well, so we're not doing any injury other than the injection itself. So it's possible that we need to combine injury or combine some way to activate at least a limited inflammatory response to do that. On time points and kinetics, that's difficult to do in sort of an in vivo screen like this. We can see when we can first observe proliferating cells, and those time points were not far off from damage time points. So seems to be following the same pathway, just not. I don't know that I can really give you a kinetic parameter. That would be difficult in this setting.
That's really impressive. And you know, the immune system keeps coming up and sort of inflammation, right? So I think we're all going to have to start getting our expertise in immunology and cell autonomous and non-cell autonomous responses. So it's really beautiful. Sarah, okay, so you know, fascinating work on photoreceptor maturation. And as you know, the photoreceptor synapse is assembled in a stepwise manner, right? So horizontal cell neurites are the first to interact, and then bipolar dendrites are inserted. And this is something that we discussed a lot in the AGI meetings. You know, do you have thoughts on that process? And I know you're not really getting horizontal cell processes as part of your ex vivo system, but is that something you guys have thought about bringing in? Is it something you think is important for studying this ex vivo? Really impressive with the imaging you were able to do, and it'd be really powerful to start to study that in an ex vivo system. So any thoughts on the horizontal cell contribution?
Yeah, one thing that's kind of interesting about sort of horizontal cells is I know there are some papers from like, I think it was like the 80s or 90s that found that you could just ablate horizontal cells and get outer plexiform layer formation that looked totally fine, which, you know, would kind of suggest that maybe they're not necessary. But that seems like, you know, I don't want to say they're not important because otherwise why would they be there? But yeah, it would be awesome to be able to like co-culture specific, like purified photoreceptors in horizontal cells. One big challenge, so our cultures aren't pure, which is one reason that the high content screen results were a little hard to interpret. And that's because purifying these populations is a bit of a challenge. And specifically with the photoreceptors at the ages I'm looking at, so at day 40, photoreceptors make up about 1 in every 10,000 cells. So just the technical challenge in getting a purified population, it's just not reasonable right now. So yeah, that would be awesome. I guess that's
tricky. They're really rare.
Yeah.
It's hard to get them. Yeah, fantastic. All right, so let's go back to Sahil, if y'all don't mind. We still have about 20 minutes, Steve, is that right? Let's see.
Yep, that's right, till 2:30.
Okay, fantastic. So Sahil, can you maybe comment on the mitochondrial transport associated proteins after optic nerve crush and tell us a little bit more about your thoughts on that?
Sorry, yeah, it's a little hard to determine exactly what that means. We do know that previous work has shown that mitochondrial-associated proteins in the peripheral nervous system do kind of increase in their transport after injury and related to regeneration, while those in an injured central nervous system don't. And there are regulators like ARM-CX1 that have been shown that to be related to increased mitochondrial transport in a regenerative CNS system. So we think that KIF5A as a molecular motor, and it's known to be associated with several of the mitochondrial related proteins in biochemical studies, as well as in our study. We think the lack of KIF5A and other anterograde motors is one of the major reasons why mitochondria and mitochondrial related proteins aren't transporting and they're affecting just general mitochondrial trafficking in these regenerating axons. So if we can restore KIF5A, which we showed in vitro, but we haven't been able to get the compartmentalized transport from the cell body to the axon in vivo. If we can get that component down during an injured optic nerve, we might be able to get more mitochondria trafficking towards the site of injury and towards the axonal compartment.
Fantastic. All right. Okay, so Jane, there's several questions. Let me just go back. Sorry, too many pieces of paper here. You know, I guess, you know, there's a therapeutic angle, clearly, Jane. And, you know, one question came up regarding PTEN, right? And it is a tumor suppressor. And, you know, if you think about, okay, would you want to target this pathway? And you'd want to be very careful about targeting this pathway, not to induce hyperplasia or some sort of malignant state. Have you thought about that? And, you know, thinking about therapeutics down the road, would it be largely local delivery, right, to sort of avoid these sort of off organ effects or off target effects. Any discussions within the lab about how that might eventually move forward clinically?
So we haven't talked too much about how this will translate clinically in terms of what you discussed. However, I think that what the research shows is that any sort of global ubiquitous delivery of, for example, P10 can have detrimental effects because it's not just involved in one thing, and that could certainly be the case with our gene targets. So I think that any sort of discussion and thinking about how we would use these genes and address anything at the clinical level, like for example, glaucoma, we would need to have really kind of a laser view, really get to the cells that are involved in glaucoma and really don't affect anything else. But, that's something that we kind of have to think about more deeply than the answer that I gave you.
Yeah, and I'm always impressed when I hear about some of the therapeutic approaches in the vision community, just how much precision there can be for delivery, release, these sorts of things. So, people always talk about the visual system as an accessible part of the brain. It really is quite remarkable in this sense that you can do a lot there where you couldn't do it in other regions of the central nervous system. So fantastic. Okay, so Konstantin, you really made a powerful case for the use of this rat hippocampal neuron system, right, to complement the retinal ganglion cell work. So I thought maybe, just sort of commenting for the audience on, at what point do you want to do the sort of retinal ganglion cell specific assays, and when do you take advantage of the hippocampal neuron assay, and how do you sort of cross-validate those? I thought it was a really elegant approach.
So the reason we choose to go with hippocampal neurons is just, a technical. We could not get a reliable cell isolation and viability of mouse RGCs in culture to do this kind of test. And our collaborator, Vance Lemon's lab, they do hippocampal neurons routinely. So they use all the they have all the necessary machinery and techniques for doing that. And so we just, accepted this way of testing. And, now we're, after we got the results, we're transitioning into mouse.
Beautiful. Yeah, no, really elegant. And then also you had used a Bax knockout mouse, from what I recall from some of the discussion.
Oh, yes.
Can you comment about why that's so important for these studies?
Well, our HB9 Bax knockout mouse was the mouse that is showing on-off direction sensitive cells. And in order to see if thrombospondin has effect on IP-RGCs or direction selective cells, we decided to go with this mouse model. And there was no result. So it means that thrombospondin does not specifically affect on-off direction selective cells. We also tested other potential subtypes of RGCs by just using a regular mouse and performing the optical nerve crush, injecting thrombospondin 1, and then giving the mouse AAV mediated expression of BCL2, which is anti-apoptotic protein, to prevent a potential apoptosis of other cells sensitive to injury. And we did some regeneration in this kind of experiment. So it means that thrombospondin 1 does not only induce regeneration of axons in IP-RGCs, but also other subtypes of RGCs.
Beautiful. Thank you so much. All right, next question is for Thon. It's about ATAC-seq, and the question is, when you look at the ATAC-seq data from the regenerating models, were there common regulatory elements that were in some of those very selective peaks? And just from some of our work in the single cell ATAC world, I've been really impressed with some of the specificity of those data and those peaks. So have you guys started to mine that, or is it too difficult to pull out really key transcription factors just from the ATAC-seq peaks.
So one thing I have to clarify that because we compare like fish and chicken and mouse, right? So all ATAC data we did only on the fish and on the mouse only.
Okay.
Because in the fish, it's not possible. In the chicken, it's not possible to isolate the molecular cells. But even, I mean, although now you can do single cell ATAC-seq on that, but when we did that like 2-3 years ago, we try to do both ATAC-seq on that, but we couldn't get the isolated molecular cell to do both ATAC-seq on that. So that's the first part. So for the, is it any common sequence of the regulatory between fish and mouse? So, I mean, consistent with other groups reported that many of neurodegeneration factor that we activate in the fish during the injury, they already open in the resting state already. So they all open in the fish, but they always close the mouse, like ASCO1 or NEI28, they always close. So so in the fish somehow they only prone to open, but not in the mouse. So yeah, so now we are working toward try to overexpress the factor that increase in the fish, but not in the mouse, try to push the mouse to become a neuron.
And another quick question was, do you have to knock out all 3 of the NF genes? I guess it's NFI, A, and X to get the phenotype.
So yes, the quick question is we don't know. We based on our previous study that when you look out NFI factors, because they're very, very similar binding site, and when you look out individual of them during development, it have an additive effect on like proliferation of the placental cells. So we see if you look out all ABX in the placental cells, they have like a prolonged proliferation. So they definitely have additive effect there. And in NFI, knock out molecular cell out of molecular cells. If you look at by NFI only without any injury, the molecular cell only losing the characteristic. I turn on some AFCL1, little bit AFCL1, but they don't do anything until you injury them. So we think we have to do lock out 3 of them to do any good stuff.
That's impressive. Wow, thank you so much. All right, so James, there's a question. It says in the C6 microexosomes, exosomes, got that right. Is that a heterogeneous population, or are there even tools to study that? So if you have a population of these exosomes, how do you know how different they are from each other? Can you further refine that? Sort of what's the state of the art in the field?
Yeah, really good. And the field is actually sort of changing all the time. And as I indicated, the original screen was done with a mixture, a very heterogeneous mixture of different vesicles. When we then use some more density gradient purified ones, we have what are traditional exosomes, but even in that population, there is some heterogeneity, and there may even be non-vesicular components in there that are being transferred between cells. For the gradient purified, we think that's mostly then the activity is from membrane-bound small vesicle, small extracellular vesicle, and that was most consistent with the data. But that's also surprising. One would have guessed that some of these larger microvesicles would be maybe even better. It's very rare to have a full-length messenger RNA in a small exosome. So you wouldn't be delivering mRNAs. You'd have to be delivering protein cargo or small, like microRNAs. So in some ways, it might have been a first guess that these larger microvesicles would be more robust. But the activity we got from the C6 was from the small EVs, and not that heterogeneous, but still some other particles in there.
Fantastic. Yeah, no, it's a rapidly changing field. That was one of the things I came to really appreciate from participating, and it's exciting. Are there efforts to really kind of profile normal cell populations and get a deeper understanding of which ones are primary contributors to this sort of exosome world that we're learning so much about?
Right, good question. So we know all cells are secreting these vesicles, and probably the worst answer is that it seems to be all context specific. So it's going to depend on the cell type you're looking at. It's going to depend on a variety of factors. And for instance, if you stress cells, you get a different population of extracellular vesicles. If you get things like ionomyosin or oncogenic stress, you get a different population. So that's going to be difficult to parse. And probably the most frustrating thing is from publication to publication and lab to lab, that's going to be difficult to parse because of exactly how the cells are cultured, how the EVs are isolated, and then how they're finally purified.
Yeah, and I give the field credit, right? Trying to standardize not only just the nomenclature and what you call things, but how you purify the protocols. And you guys have really been a leader in that. So, you know, that's absolutely essential for rigorous science. Yeah, great job there. Thanks. All right, so Sarah, back to your really interesting observation and the difference in rods and cones, right, in this sort of neurite outgrowth phenotype. So the question is, are you extrapolating that the cones have the dynamic growth cones at day 40 since your NRL-GFP rods do not show any dynamics? I think that's what you concluded at later time points. And then did you specifically purify the cones, right, using your fluorescent reporter to show that in a more purified culture? And maybe do the rods influence the cones and vice versa, these sorts of interactions?
Yeah, so I do assume that the younger photoreceptors are all cones. So that CRX-TD tomato reporter line should be all post-mitotic photoreceptors. But we also know that rods don't start being born until just before day 70 in these human retinal organoids. And so I just kind of assume if it's a photoreceptor and it's not a rod because rods aren't born yet, then those are cones. But I haven't tried this with a cone-specific reporter, though the GAM lab has developed one and is working on getting a dual reporter line so I can look at like both rods and cones at the same time, which would be really interesting to see them sort of side by side and have that really great control of the same differentiation parameters. Because I do think there's certainly some slight variation in developmental times. Was there any other part to the question?
Well, I guess I have a follow-up. And you know, many years ago from my time in Connie Sepko's lab, one of the lessons I learned, not from my own work, but from just sort of historical work, was that rods really care a lot about the density of rods in a culture system, whatever that might be, right? So how much did you guys play around with cellular density to see if there might be extrinsic factors that are contributing to some of this?
Yeah, so I didn't play around with density, especially in terms of like cell type specific density, because again, the challenges of trying to purify was just too difficult. But we did play around with just like, you know, how many cells are we putting down on the plate from the whole organoid? And certainly like the more cells you have, the more sort of like motility I think you just kind of end up with. And so you definitely like higher numbers of cells in the dish, you're going to get photoreceptors, like more of them kind of getting stretched out. And actually a lot of my experiments, I've been doing lower densities specifically so I can look at those terminals in isolation, not on top of other cells. So a lot of what I'm doing is sort of like, that's a good thing to keep in mind. And
yeah, that's cool.
Yeah.
So I think that's going to wrap us up because we don't have time to go through one more round. I will just, you know, close out and put in my 2 cents, which is this has been an incredible initiative from the NEI, as you all can see from this remarkable research. It's very different, in my view, from standard R01 type funding mechanisms. This was high risk, high reward. I love that the NEI did this. I hope they're going to continue to do this. I don't want to put Steve and Michael on the spot, but you got my vote if you all want to keep this up, because I think this kind of high risk research is really what the field needs. So thank you for letting me participate. It's been a pleasure.
Thank you.
Thank you.
Thanks, everyone. Great job.
Thank you.